I'm Shama Mirza. I'm an assistant professor in the Department of Biochemistry and Biotechnology and Bioengineering Center. My work is mostly on mass spectrometry, and in BBC we have an excellent facility of mass spectrometry. So without the availability of these instruments, I think the protein characterization experiments and also the technology development that we are doing is not possible. And in addition to that, I would definitely like to add that the bioinformatics group we have in BBC is excellent, and they made some unique tools, and without those, the data analysis of the huge mass spec data that is generated from the high throughput instruments is not possible. And of course, BBC has an excellent faculty and also the staff who are very helpful, and they're very friendly, and I could find several collaborations within the center and also within MCW and outside MCW with their help. We are interested in identifying the differential protein expression under hypoxic conditions in angiogenesis. And we do this in unique rat model systems that have different hypoxic angiogenic behavior under hypoxic conditions. Those are the BN and SS rats. And we culture rat endothelial cells and look for the differential protein expression using mass spectrometry. And in this process, we are developing new technologies so that we can do some precise protein characterizations and identify the proteins that are expressed in a different way, and also identify the modifications, post-translational modifications in these cells. The main focus is technology development in our lab. We are coming up with new strategies, improved strategies for relative quantification of proteins, and also we developed new methods for identifying post-translational modifications also. And these methods are better than the traditional methods because these are unbiased, and you can identify the modifications that are not known, like even without having a prior knowledge of what kind of modifications you are looking for. This method, the new strategy that we came up with, can identify the modifications that are not determined earlier. So another focus of our research is Proteus syndrome, and this is a very rare disease, but with severe segmental overgrowth of variety of tissues like skin, muscle, blood and lymphatic vessels, and so many other tissues that are overgrown. And we are interested in finding differential protein expression in these syndromes too. And the interest is if you can find a causal agent for that, you can find the therapeutic agent for this. And so far, to our knowledge, there are no known studies done in Proteus syndrome, proteomics of Proteus syndrome. We are doing the hypoxia-induced angiogenesis and Proteus syndrome, and we established an atlas of proteins in vascular endothelial cells for hypoxia, and also we found some differential protein expressions in Proteus syndrome, and we are in the process of characterizing these proteins further, and we need to validate these further. And coming to the technology development, we found an improved method for relative quantification of proteins using mass spectrometry, and this is a fast method, and it can we could overcome the limitations that have been there in the O18 labeling methods like back exchange and extensive sample preparation, and all these kinds of limitations we could overcome. And with the new strategy for the post-translational modifications, as I told you, it's definitely better than the existing ones because you can identify more number of modifications within a simple single mass spec run. And so this is definitely useful because especially when you have very limited sample amounts like clinical tissue biopsies, so you can finish all the analysis within one mass spec run, and you can analyze the data.